Hey everybody, welcome. Proteins, we're here at 5.4. This is the biggest section of this biggest chapter in the textbook, 10 pages, all the way in the 10th edition anyway, from the page 75 to like page 83 or 4. It's a long, big section, but there's a lot that's important here, and proteins are really the most important things in your body. They do everything. Lipids are fine, carbs are fine, nucleic acids are fine, but it's the proteins that help us see, help our heart to beat, help nerves to conduct impulses. Everything that you do comes down to proteins. In fact, there are about 200,000 different types of proteins that we have in our bodies. What we normally do is we eat proteins we get proteins from food, and then we break down the long polymers into individual monomers, these individual amino acids. And then with the help of language on DNA, it tells us which amino acids then to take from that pool of, say, trillions of amino acids and start to put them in our own sequences so that they can fold and then do something in our body. If we were to take you and tape you down to the Weckmerd playing field across the street and have one of those fans blow on you for 6 months, and come back 6 months later and you're just a pile of dust, and we were to look at that dust, 50% of that would be proteins. They're super important. Many would argue that they are the most important things that we have in our bodies to help us survive, even more important than DNA. So let's dive right in. Make sure that you pull this out, because the essay prompts for the next test are going to be right here, and I'm going to say, hey, this is an essential knowledge that the College Board wants you to know and be able to demonstrate. And so I'm just going to put that as a prompt on the test, and then you can just write and draw and show me that you understand what the prompt is asking or saying, or what you're supposed to be able to provide as far as knowledge in that case. So go ahead and take a look at this. I like this little summary that they have for all of these different macromolecules of our chapter. I'm going to read this here. It says, proteins have many structures resulting in a wide range of functions. You can see here, here's the general structure of an amino acid, and we'll get into all the nuances of what you see there with the amino acid monomer. Do you need to memorize all these? And the answer is probably yes, at a minimum, to be honest with you. Not all of them, but the ones I'm about to touch on, these are the most important examples of proteins that organisms have, and then the functions that they have. And we're going to see them again and again all throughout the year. So I think it's all good. We're just trying to set the table for later in the year when we pick up the pace and things get a little bit harder, that I know that you have all this baseline knowledge. Well, everyone knows what an enzyme does. It helps to speed up reactions that would have ordinarily taken place on their own over a longer period of time. You can see here is a picture of this enzyme called sucrase. At the end of chapter 6, there's a bunch of stuff on enzymes, so we're just going to touch on enzymes here. But you can see that something goes in, the green thing goes in, that would be the substrate, which is the table sugar sucrose, and the enzyme is going to break the table sugar into its two monomers, glucose and fructose, probably through a hydrolysis reaction. You can see that water is being added. The idea behind enzymes is that they can do what they do many, many times and not be broken down themselves. So they're just facilitating reactions, and then they're ready to facilitate the next one. And some of them work thousands and thousands of reactions per second, like tens of thousands. It's crazy speed. So enzymes are important. Something else that I see up here are support proteins. Sorry, these would be like ligaments, tendons, hair, nails, skin, things like that are made up of proteins in our body. Here's an example of my friend Sophie Russell's split end from last year. You should let her know that you saw this. Transport substances are things like these proteins that exist in cell membranes, that like channel proteins or carrier proteins. They help shuttle stuff through membranes that are most often very polar, so that they couldn't go through, or large or charged, they couldn't go through the middle of the membrane on their own if it was just trying to diffuse right through the membrane. So they need like a tube, like a protein there. Hormones are important. Some examples of protein hormones would be like insulin, glucagon. We've talked about those. Oxytocin, which helps in uterine muscle wall contraction in women, as well as it's released. It's the cuddle hormone. So if you ever have oxytocin released, you're in love with the person who you're around, essentially. Receptor proteins, let me show you an example of that. Here's a receptor at the cell surface, and what I want you to understand here is that it has this very, very unique shape. You can see it, it's all these blue bumps and everything all over it, but with proteins, shape equals function, or structure equals function. So every one of those things is super intentional, no coincidences. You can see now that this thing is fitting like a key into a lock. And what that's going to do is it's going to cause something to occur, probably reactions down below inside the cell. We'll learn more about that in chapter 11. But the point is that it's super duper specific. And we can draw other cartoons if we like to show the specificity here. We'll get into that detail in minutia in just a moment. Finally, antibodies are really important. They help protect us against diseases. We'll get into that. That's chapter 43, I believe, is the immune system. So we'll get into that in the second semester. Well, a protein consists of one or more polypeptides. How many do you see here? Let's take this moment to pause for a second. What I have up here is the central dogma of biology. This is what Watson and Crick called it. Watson and Crook called it, and we'll get there later. What is made from the message that exists on DNA? I've kind of drawn this little cartoon representation of DNA here, and this information is used to make something. Does anyone know what that is that's made? Well, it's made in the nucleus, and just look for a different color. That's called RNA. There's lots of different types of RNA. We'll get into that in chapter 17. But this arrow right here, go ahead and say it with me. You're right, transcription. Good job. And then the message on RNA is translated, that's right, into a sequence of amino acids. So I've just written up there AAs, and that's going to be my abbreviation for amino acids. You can see it down here, AAs. And then this is going to go towards that happens in the endoplasmic reticulum with the help of ribosomes, or maybe some free ribosomes. But the point is that then this thing folds most often inside the Golgi body, sometimes in the cytoplasm. And then what we make are proteins. And that's how proteins are made. Yes, you have to know this backwards and forwards. And we'll get into a lot of the detail, more specific detail here as we go through the chapters. Well, polypeptides are just many amino acids that have been put together. There are 20 different amino acids that exist out there. There are many more than that, but 20 that we use, for instance, and most living organisms use. 9 of them are essential, which means you have to get them in your diet. But 11 of them we can actually create. These amino acids just from atoms that we get from foods, and then we have that amino acid available. If the DNA message says, hey, by route of making this message, hey, make this sequence of amino acids so I can fold it into this certain protein. 11 of those we can make on our own, those amino acids, but 9 we have to get in our diet. So look for essential amino acids in the foods that you eat. I have my trusty cereal box here. And it does say that it has 2 grams per protein per serving. Honey Bunches of Oats. Well, this is what a representative amino acid looks like. And you can see that it has 4 component parts here. It has a carboxyl end on the right-hand side. You can see an amino end on the left-hand side. It has a central carbon bound to a hydrogen, and then it has the R group. What the R group is it's the rest of the amino acids. So amino acids always look like this, and then the variable portion is the R group. And that R group gives the individual amino acid, like the 20 that we're going to study in this class, its character. These most often are either hydrophilic, hydrophobic, or ionic, which means they're charged at pH of 7.4, which is the general pH that you find not in the lungs or the digestive system, but most often in cells in the bloodstream. I'd like you to draw this. Press pause. I think it's that important. Go ahead and draw this and then identify the carboxyl end of the amino acid, the amino end of it, and the R. Well, you can see up here the R group is important, and we'll get into how that infers character on each of these amino acids. I want you to find this picture on your handout or your textbook. And what this is trying to show you is that individual monomers can be put together to make a polymer or a peptide, polypeptide of amino acids. These are called peptide bonds. That's why I didn't define this earlier. It's just called a peptide bond. It's a covalent bond, similar to what happens with carbohydrates and then also nucleic acids. When these monomers come together to make polymers, you can see here it's a dehydration reaction. See it there and there. So how many peptide bonds are used when you put 3 amino acid monomers together? 2 peptide bonds. This is called a tripeptide. This is a dehydration reaction. We've seen this before. Is this important? Press pause again. Draw it, please. And I'm going to have you draw even more stuff today. So I'll be quiet. Just note that there's an amino end. This is where actually the protein starts. So I'll just write the word start up here. This is where it begins. The message on the DNA, which coded for all these other messages that were translated and said, all right, start making a sequence or a chain of amino acids. Amino end is the beginning of that, and this is the end of the amino acid chain. I get it that it's opposite of what you see on the screen, but just imagine if you were to go out in the hallway and look through the board and through the wall and through the board, it would look exactly like what you see on the screen.
In order to understand peptide bonds, let's cover a few key concepts. Proteins are a primary building block of life. A protein is a compound made of small carbon compounds called amino acids. Several covalent bonds called peptide bonds join amino acids together to form proteins. A peptide bond forms between the amino acid group of one amino acid and the carboxyl group of another. In this example, the acid group of glycine joins with the amino group of alanine. The result is glycosalanine. The acid group of glycine loses an atom of hydrogen and an atom of oxygen. The amino group of alanine loses an atom of hydrogen. These atoms combine to form a water molecule. More peptide bonds can form in the same way, creating a chain of amino acids called a polypeptide. Polypeptides are essential to human life, but do you know where you can find them? Polypeptides are found as proteins like keratin in hair and nails, casein in milk, and hemoglobin in the blood. See how important they are?
Okay, so if you were to take this essential knowledge sheet from the College Board, which I've just put in a Word document, and look down below, you'll see this one right here. And this could be a potential essay prompt for your exam. You just have to be able to draw this. Use this as your prompt. Draw me something, write me a paragraph of what you're drawing, and so just keep it simple, but read the book, and here I've given you some information that should help you be able to demonstrate knowledge of what this is saying students need to be able to do. Well, once we start putting these things together, you're seeing here, like this is, I guess I'll point at the screen. You can see here there's the amino end, and then there's glycine, isoleucine, valine, cysteine, glutamine. And there's a lot of amino acid here in a long chain. And that's fine. A polypeptide is great, but it doesn't really do anything. What has to happen is it needs to fold. And you can see here that it's starting to fold. This is a very unique sequence, and it's dictated by the letters on DNA. Well, here are some examples of amino acids. These are polar amino acids. Now, you'll see this picture in your textbook. They'll have all the amino acids here. Do you need to memorize all these? The answer is no. You can always access them. I've given you some on the handout as well. You can always access them somewhere else. But do you need to know the differences between the polar, nonpolar, and ionic amino acids? Absolutely. You need to know what ramifications that has for protein folding and some of the general characteristics. But don't memorize these individual structures because you can always look them up. That would be stupid. Here's something that a student told me a few years ago is that it snows at the poles. And so this is how she remembered that whenever she saw a sulfur, nitrogen, or oxygen atom on the R group. So understand what you're looking at here. Every one of these 6 amino acids has an amine group, a central carbon bound to a hydrogen, a carboxyl group, and then the white box is representative of the R group. That's what is different in each one of them. And if you see one of those 3 atoms, they are going to share electrons unequally with the hydrogens that they're nearby because they're super electronegative. We've been talking about this for weeks now. They're going to create hydrogen bonds. They're going to have these little electrical dipoles, like more positively or negatively tended portions of the R group. And that's going to cause them to kind of be attracted or repelled from one another. It just gives them character when the thing starts to fold. Well, here are the nonpolar ones, and this is exhibited, as you can imagine, by tons and tons of hydrocarbons, tons of carbons bound to hydrogens. One of them has an S in it, sulfur atom, but it's buried. Another one has a nitrogen atom, but it's also buried. It's not kind of near the end, and there's not many of them. So just generally speaking, tons of hydrocarbons. You can imagine that these things will be hydrophobic, which means they don't like water. So when the protein starts to fold, you know, life exists in an aqueous environment, and so they're going to try to get buried in the middle of the protein. They're going to try to get away from kind of the watery areas on the outside of the protein, and that's going to help rigidify the protein as far as certain structure that is made. Well, these electrically charged ones, you know what an ion is? That's an atom that has either gained or lost an electron, and the idea is that at certain pHs, specifically the body pH, some of the carboxyl groups gain, the oxygen in the carboxyl groups gain an electron. I don't know why, but they now have a charge. They literally have a charge, a negative charge. And then some of the amine groups in the R groups will gain, so I guess that would be lose an electron. And so now they have one more proton than an electron on the nitrogen atom. And so the point is they have a positive charge, like they literally have a charge. And you know, like charges repel and opposite charges attract. And you're going to see that once these big polypeptide structures begin to fold. You're going to see these characters come into play. Well, if you look at this one, number 2 here, let's read it. The R group of an amino acid can be categorized by these chemical properties, and the interactions of these R groups determine the structure and function. I just went over this. You could draw me an example protein on the test. You could talk about these different types of R groups and how they will cause the folding to occur in different ways. So spend some time with this for sure, please. Well, let's take a moment, press pause, and just go over, take a breath and go over what you've learned so far. Well, here's a picture, a cartoon picture of a representative protein, and you see all these squiggly lines here. That's just supposed to be, let's say, hundreds or thousands of amino acids in sequence, and they have folded as a result of the rules that govern protein folding due to the characteristics of these different amino acids because of the different types of R groups. And you can see that this thing has a super specific shape. It has a groove which does something. Either it's going to transport stuff or it's going to act like an enzyme or whatever. You can see here that a 3-dimensional structure is really important. Here's an example of the outside of the flu virus, which has protein coat on the outside, and then we would make antibodies to that in order to immobilize those flu viruses, and it's all super specific. If one of those amino acids is in the wrong place, you can imagine that it would throw off the entire shape of the protein. It would kind of like kink inwards where it shouldn't or whatever, and it wouldn't do its job. So protein structure determines its function. That's a super important thing. That's the same for everything in biology, but most specifically with proteins for sure. Well, functional protein often doesn't have just one polypeptide. It has, the example I showed you earlier had 4 polypeptides. They will then come together in order to create like a super shape in order to do something. Like the example I showed you earlier was hemoglobin, which carries oxygen on the surface of red blood cells in our bloodstream. And that's a protein created with 4 polypeptides. We can see this little kind of ball model. The white balls are hydrogen atoms, the red ones are oxygen, and then the blue ones are carbon, I'm pretty sure. And you can see then there's different ways to represent the portions of a protein. I guess I'm second guessing myself. That's how it usually is when we build these like ball and spoke models with plastic stuff, but that would be way too big per atom. So I think that those are representative of different amino acids. Well, let's go to page 80 in your textbook. Go there because this, I'm gonna, I used to have this genetics professor at Cal Poly when I was an undergrad, and he would always draw this man, upside down man. I remember this 20 years later, like this. And he would say, and I don't have a very good Indian accent, he was from India. He would say, I would stand on my head to make sure that you understand this this concept, and I always remember the upside down man. I don't know why that is, but this is like super important. Spend 10 minutes on pages 80 and 81, and then tomorrow do 10 more minutes, and the next day do 10 more minutes, and so on. It's kind of a summary of all this stuff, and it is super important. So I've given out huge portions of the exam before on these 2 pages, and I've seen this on the AP test itself in May, so please focus on these 2 pages. Well, there's 4 levels of protein structure. Once this polypeptide starts to bend and stuff, and we're going to talk about primary, secondary, tertiary, and quaternary protein structure as the thing begins to fold. And there's rules for all these things, and I'm about to show you what they are. Remember, the primary structure is just the sequence of amino acids. It's governed by these 3 letters that exist on RNA as a result of DNA. So the idea is this. So there's tons of DNA, and there's tons of 6 billion letters of DNA per cell. But there are certain specific portions of DNA called genes which dictate RNA, which sometimes most often makes proteins. That message is used to put amino acids in sequence to make proteins. And the idea is that every 3 letters on the DNA corresponds with 3 letters on the RNA. We call that a codon. And if you remember from 10th grade biology, the ribosome comes in, it reads the codon, and then a transfer RNA recruits the proper amino acid to come in sequence, and then we can start adding them together with these dehydration reactions and create these peptide bonds, and then all of a sudden you start to create a long chain of amino acids. We'll get into all this detail in chapter 17. We'll go over it in a lot more detail than this, but this is just the basics here. This is called primary structure. It's just a line. It hasn't really folded yet due to any characteristics of the R groups. It's like the order of letters in a long word, and it's super important. Make sure that you understand the central dogma and how the information flows from DNA to the creation of proteins. That's super important, because you can imagine if you're laying out at the pool at Singletree all summer and you have a photon of UV radiation which changes an atom in one of the letters on your DNA, and now all of a sudden it's a different letter, then the RNA is going to be different, the amino acid put in sequence is going to be different, and your protein is not going to fold correctly. And now all of a sudden you can't control, let's say, cell division as well as you used to be able to in this cell. And then so the cell starts to go haywire and divide all the time. And then all of a sudden you have a lump or a weird color thing on your skin, and you may have a lot of trouble because of that. But that's how it happens, is that DNA dictates RNA, which dictates the sequences of amino acids. Well, let's go here. Secondary structure. There's 2 types of secondary structure. You can see it on there: alpha helix and beta pleated sheet. Understand that secondary structure, this is super important, it results between from hydrogen bonds between carbonyl and amine groups of the polypeptide backbone. So here's what we're talking about. As these things start to fold, and so I've drawn this squiggly line up here, as these things start to kind of fold next to each other, sometimes they'll form like these things, these spirals, or these, it's called the beta pleated sheet, and it looks literally like an S or a sheet here. And the idea is that there's sometimes interactions between the amino acids that are near one another. But this is super important in secondary structure. Write yourself a note. it's not due to the R groups. Not yet, that's tertiary structure. Secondary structure, it's due to just the things that exist in every amino acid. These carboxyl groups have carbonyl, it's a C double bond O is a carbonyl functional group, and you can see there that it's creating a hydrogen bond with a totally different amino acids amine group. And then with respect to alpha helix, it's I think every 4 in the right spot, and you'll see because it's not an alpha helix everywhere, but where it happens, I think it's like every 4th amino acid you get this hydrogen bonding between the hydrogen atom in the amine group and the oxygen atom in the carboxyl group, which would be a carbonyl C double bond O. Understand that there's like kind of a positive and negative tendency or a dipole or portion to those portions of the amino acids, and then they kind of are a little bit attracted, and you've seen hydrogen bonds before. So there's 2 types here, beta pleated sheet, alpha helix. Not the R groups. I'll show you a movie on each of these. Here's our first movie on the alpha helix.
The alpha helix is one of the most common secondary structures in proteins. Amino acid side chains project outwards from the polypeptide backbone that forms the core of the helix. The chain is stabilized in this conformation by hydrogen bonds between the backbone amino group of one amino acid and the backbone carbonyl group of another amino acid that is 4 positions away. These interactions do not involve side chains. Thus, many different sequences can adopt a helical structure. Alpha helices are regular cylindrical structures. One full turn occurs every 3.6 residues and extends the length of the helix by approximately half a nanometer.
Okay, here's a little movie on how the beta pleated sheet is created and why it's important.
The beta sheet is another common secondary structure. In contrast to an alpha helix, it is formed by hydrogen bonds between backbone atoms on adjacent regions of the peptide backbone, called beta strands. These interactions do not involve side chains. Thus, many different sequences can form a beta sheet. A beta sheet is a regular and rigid structure, often represented as a series of flattened arrows. Each arrow points towards the protein C terminus. In the example shown here, the 2 middle strands run parallel, that is, in the same direction, whereas the peripheral strands are anti-parallel. The amino acid side chains from each strand alternately extend above and below the sheet, thereby allowing each side to have distinct properties from the other. Beta sheets are usually twisted and not completely flat.
So I just want to clarify this or repeat what she said. If you have beta strands that are going, so the arrows always point to the end of the amino acid polypeptide. It could be 1,000 amino acids long, but it has to have a C double bond OH at the end. And so, I'm not going to go through here and do this right, but the point is you can imagine that there's arrows pointing different directions here towards the C terminus. And if you see a sheet here where the arrows are pointing in the same direction, that's called parallel, but I'm going to erase this, and then I'm just going to draw an arrow the other direction. That's called anti-parallel. You can see that here in this little picture that's now up on your screen. You can see these alpha helices and beta pleated sheets. Well, to further fold the protein and rigidify the structure so that the protein can do whatever it needs to do in the organism, there's tertiary structure. And this is the interactions between R groups. Remember, these R groups are either polar, nonpolar, or ionic. They're going to have a tremendous amount of character and cause lots of folding to happen. So here's tertiary structure. It's not the interactions between the backbone constituents like secondary structure. If you think I'm going to ask you this on the test, it's super important that you understand that distinction. Well, this is a picture that I like. It's on in the 10th edition of Campbell. It's on page 81. You can see it down here. And this talks about 4 different types of R group interactions in tertiary structure of protein folding. So here's our 4 types of interactions between R groups. First one's called van der Waals. These are just hydrophobic interactions. All it means is that on the inside of the folding protein away from the aqueous environment. These hydrophobic amino R groups on these amino acids that have tons of C's and H's all over them, they kind of just like to be near one another. Van der Waals, you saw that earlier in the year. I told you it wasn't very important. This is the only place you're going to see it for the rest of the year. Up there in the top left corner are hydrogen bonds. You know that that's caused when there's unequal sharing of electrons in a covalent bond between an electronegative atom like a sulfur, a nitrogen, or an oxygen, and then a hydrogen near it. It's going to cause these kind of partial positive and negative charges that are going to line up and create a hydrogen bond. You can see that. Disulfide bridges are really important. Cysteine is the amino acid that we use most often in creating these disulfide bridges. They're super strong, and they really help rigidify structure. And then finally, down there at the bottom, ionic bonds are also strong. You know that opposite charges attract and like charges repel. And you see those on R groups of ionic amino acids at the correct 7.4 pH. Well, if we start to put multiple polypeptides that have folded in tertiary structure together, we can then get quaternary structure. This is the final type of structure, and all this just means is that when you get multiple polypeptides that have folded together, it creates kind of this larger structure to create a functioning protein. Here's a few examples from your textbook. You can see collagen, which is a protein in our hair and nails. It's a tri-polypeptide structure, and then this is hemoglobin on the right-hand side there that has 4 polypeptides.
Proteins are polymers constructed from the same set of 20 different amino acids. The primary structure is the specific sequence of amino acids specified by the gene. In the secondary structure, portions of the protein begin to fold in neat three-dimensional formations. 2 kinds of secondary structure units are the alpha helix and the beta-pleated sheet. Folds and coils of the protein are the result of hydrogen bonds that occur at regular intervals. The tertiary structure is formed by interactions between side chains of various amino acids. At this stage, some proteins are complete. Other proteins incorporate multiple polypeptide subunits, creating what is known as a quaternary structure.
Proteins play countless roles throughout the biological world, from catalyzing chemical reactions to building the structures of all living things. Despite this wide range of functions, all proteins are made out of the same 20 building blocks, called amino acids. The way these 20 amino acids are arranged dictates the folding of the protein into its unique final shape and its function. Amino acids are made of carbon, oxygen, nitrogen, hydrogen, and sulfur atoms. These atoms form an amino group, a carboxyl group, and a side chain attached to a central carbon atom. The side chain is the only part that varies from amino acid to amino acid and determines its property. Hydrophobic amino acids, such as leucine and isoleucine, have carbon-rich side chains which don't interact well with water. Hydrophilic amino acids, such as serine or threonine, interact well with water. Charged amino acids like glutamic acid or arginine interact with oppositely charged amino acids or with water. The primary structure of the protein is the linear sequence of amino acids as encoded by DNA. The amino acids are joined by peptide bonds, which link an amino group and a carboxyl group. The water molecules release each time a bond is formed. Specific amino acid sequences give proteins their distinct shapes and chemical characteristics. These protein chains often fold into 2 types of secondary structures, stabilized by hydrogen bonds. A protein chain can fold into a rigid alpha helix, forming regular patterns of hydrogen bonds between the backbone atoms of nearby amino acids. Backbone atoms of the chain can interact side by side to form beta sheets. Many proteins fold into a compact globular shape, with hydrophobic side chains sheltered inside away from the surrounding water. The functions of many proteins rely on this folded structure. For instance, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds oxygen. 2 or more polypeptide chains can come together to form one functional molecule with several subunits. The 4 subunits of hemoglobin cooperate so that the complex can pick up more oxygen in the lungs and release it in the body. Many proteins rely on the ability to recognize the shape of specific molecules in order to function correctly. The flexible arms of antibodies protect the body from disease by recognizing and binding to foreign molecules, and thus preventing the viral RNA or DNA to enter the cell. Collagen forms a strong triple helix that is used throughout the body for structural support. The calcium pump moves ions across cell membranes, allowing the synchronized contraction of muscle cells. The hormone insulin is a small, stable protein that can easily maintain its shape while traveling through the blood to regulate blood sugar level. Alpha amylase is an enzyme with a catalytic site that begins the breakdown of carbohydrates in our saliva. Ferritin forms a hollow shell that stores iron from our food.
Well, let's go over this one last prompt here, and then we'll be done. Specific order of amino acids in a polypeptide. The primary structure interacts with the environment to determine the overall shape of the protein. Make sure that you understand the difference between these different types of steps in protein folding. Again, take a look at in 10th edition, take a look at pages 80 and 81. You got to know it cold. I would say read it every day from now until the exam for sure. I'm going to ask you to press pause right now. Go ahead and draw whatever you need from 80 and 81. Read it. Think about it. Thanks, everybody. Have a good night.